So I get to introduce myself. Hi, my name is Ben Greenberg. I'm a Gemini. I'm originally from Houston. I now live in Dallas. And you are going to suffer with me for the next, suffer because of me, for the next two talks. We're going to shift gears. So we started off in the original sessions talking about rehabilitation, restoration of function based on what you can do. We moved into surgical and device options, and we just finished off a session on drugs that can either help augment function or drug issues that you need to be aware of that you could be fighting that are actually decreasing your function. And now we're going to shift gears and look to the future in terms of what studies are either ongoing right now or are going to come for repairing damage that's been done. The data that Mike and Lindsay talked about using a drug to restore function is different than using a drug to repair damage. So formiminopuridine, dalfampridine are wonderful at helping with function, either if we're talking about the visual system or the walking system or potentially other things, but they don't do anything to fix a broken wire or to remyelinate. And we're going to shift gears and look to what's on the horizon for this. But we're going to start off with a talk that in your syllabus and your notes is labeled What Drug Companies and the FDA Need from Us. And the alternate title that I placed on this was, so you want restorative trials in our diseases, do you? You want an investment of resources, you want focus and attention being given to transverse myelitis, ADEM, NMO, because it's through those resources that we're really going to advance our capabilities for treating you and other patients around the country. And so what do we have to do as a community to get us in a position where we are the right group to invest those resources in? And so we're going to start off with a brief introduction to clinical trials. By show of hands, how many people here have taken part in clinical research of some kind? Signed an informed consent at some point for some reason? Great. Now, in clinical research, there's lots of different studies that can happen. There are studies where we just gather data about you, and you're not exposed to any experimental agent. There are studies where we gather blood or spinal fluid or tissue from you, and again, you're not exposed to any experimental agent or paradigm. And then there are clinical trials where you agree to be exposed to some drug or intervention. And in general, those trials are regulated by the FDA, the Food and Drug Administration, in order to ensure that what's happening is appropriate. We have IRB boards to ensure that things are ethical and that they're appropriately designed to answer a question. And there are three phases to clinical trials. So phase one are in general small trials. These are first in humans. So how many people here have taken, what would be a good one, baclofen? At some point. Lots of people probably in the room have taken baclofen. At some point, there was a first human being on Earth to swallow that pill. Okay? So for every drug we take, at some point, there was a first person in line to get exposed to that drug. Any drug you take, any FDA-approved drug, somebody had to do it first, which meant it went through testing in a dish, testing in animals. There was a lot of science, a lot of work, a lot of analysis that went into before a person swallowing the drug. But at some point, somebody volunteered and said, I'll do it. Now, it usually is a young, healthy individual, sometimes medical students who just want free pizza. And you bring them in, and you say, here, swallow this. Let's see how it goes. And really, in a phase one trial, what we're looking to ensure is, number one, that people don't just drop dead on the spot. Maybe the mice were immune to something, and the monkeys were immune to something, and human beings are different. That actually rarely, if ever, happens, thankfully. And number two, we're looking to see if we can figure out dosing. So a phase one study is there purely for safety, for all intents and purposes, purely to find doses that make biologic sense. And we do a little bit of investigating to see if we get a signal of, is it worthwhile to go forward? Are these drugs going to be useful for people with a certain disease moving forward? But in a phase one trial, it's somebody stepping up to the plate to say, I am truly going to be part of that first test to determine, is this going to be useful? Once you pass a phase one study, you move on to phase two. This is a larger study. It's over a greater duration, and for the first time, we're bringing in assessments of efficacy to some degree. So we are looking for safety in greater numbers. Maybe there was an adverse event when you tested in 10 or 20 people that we just don't see, but when you go to 50 or 100 people, maybe for the first time we realize that there might be an issue with a liver or a blood count or something along those lines. So phase two is about safety, hardcore about safety, But for the first time, we really look to see, is this going to be of benefit? So while in phase 1, we have a lot of healthy volunteers, in phase 2, we're actually testing this in people who have the condition that we're interested in studying. So for the first time, sometimes we do that in phase 1, but not always. But in phase 2, it's always about the disease we're interested in studying. If the phase 2 trial goes well and the data is reviewed internally by a sponsor and by the FDA, looks appropriate, then you get a clearance to move on to a phase 3 clinical trial. And in general, this is the last series of steps before submitting to an FDA for approval. And in the United States, in order to get a drug approved for diseases, in general, you need 2 well-controlled phase 3 clinical trials in order for the FDA to review the data and say, yes, I will or not approve this for this indication. So there are 2 types of drugs in the world. There are 2 types of ways to use drugs in the world: on-label and off-label. On-label means there was a study or 2 or 3 that went to the FDA, so the use of drug X in a certain disease. So, for example, the closest cousin for us, since there are no FDA-approved drugs for the diseases we're talking about. There isn't a drug out there that on its label says, use this for transverse myelitis or NMO or ADEM. There's not a single one. So if we go to the next closest, multiple sclerosis, there are 10 FDA-approved drugs. They all had to be studied. They all had to meet the requirement for the FDA to say, this drug is approved for multiple sclerosis. If I give you that drug for some reason, I am doing what's called off-label prescribing, which I'm perfectly allowed to do. Any licensed physician, healthcare practitioner in the United States can prescribe for you anything that makes sense. The FDA doesn't restrict our prescribing. They restrict companies in terms of marketing. They restrict companies in terms of what they can say. But it's up to you and me to decide what we're going to use. Your insurance companies, however, look at what the FDA approves. How many people here have had me or somebody else have to do a prior authorization or a peer-to-peer for a denial? They are no fun. They're no fun for you. They're no fun for me. I've got better things. I don't know. I have got lots of things I would like to do with my time other than browbeating medical directors. It is worth it because we need to get the drugs for our patients, but we like to get things that are on label for these diseases to make everybody's life easier. The phase 3 clinical trials are larger, and at this point, efficacy is the primary endpoint. We are still assessing for safety, and the drug could fail to meet the requirements on either side. Either it fails to be efficacious, or there is a safety signal that is so concerning that the FDA says the benefit isn't worth the risk ratio. And in fact, the FDA rarely does that. They believe we're all adults. We can do informed consent. We can make you aware of risks. It is highly unusual for them to say that some sort of risk was too much unless it's something obvious in the trial. But it has to prove that there is a reason for you to take the drug. And this goes over a longer period of time, and the duration of the phase 3 trial depends on what we're trying to prove. So the duration, the design of the trial is purely based on the drug and what we're trying to do. So that's phase 1, phase 2, phase 3. So what are the unique features relative to rare diseases when it comes to the FDA and doing these trials? Why are we so different? So there are certain things about us that are actually quite nice, being in a rare disease community, believe it or not. So number 1 is there's less competition in general. I don't have a problem of 10 different companies beating down my door with different products for transverse myelitis or ADEM. I've yet to fight that fight. Thankfully, it's actually picking up. We have a lot more companies getting interested, but it's not at the same level. These are companies that have made specific commitments to rare diseases and are starting to ramp up their work. And then there's a law passed many years ago called the Orphan Rare Disease Act, which did 2 very important things, multiple, but 2 particularly important things for rare diseases. Number 1 is it said you don't need 2 phase 3 trials anymore. For a rare disease, you only need 1. So you don't have to do a second. That's really important. The second is it extends the patent life of the drug that gets approved, giving it certain protections in the market. So if you get approved under the Orphan and Rare Disease Act, you get a special preference in the market. There are certain tax savings and other things to companies, and this was the federal government trying to entice private industry to invest more in rare diseases, and it has worked. There are more people coming to the table because of this legislation than ever before, because frankly, there's not enough rare diseases to make the investments worthwhile without these certain considerations, and we're going to talk about the economics. And finally, and I believe this, there are certain benefits to the community that, frankly, biotech companies and pharma are well aware of. We like to talk about an us versus them. You know, drugs are expensive, especially in the United States. They're extremely expensive, and there's a lot of head scratching as to why there's the expense. We're going to talk about it. But I've actually been pleasantly surprised in my journey through this side of my career in finding a number of companies who are truly committed at the highest levels for helping out rare disease communities. They have to make it a somewhat profitable endeavor in order to get the support from their stockholders and everything else. But a lot of these folks are not looking to make a killing. These are not going to be Lipitor for Pfizer. These aren't billion-dollar investments, and they know that, and they're still willing to invest the resources coming to the table because they actually believe in the mission, for the most part. What's the disadvantages in rare diseases? Recruitment, number one. This is what scares off, I would say, most potential industry sponsors for a study. There are only so many of you. So if I take neuromyelitis optica as an example, there are between 4,000 and 10,000 NMO patients in the United States. That's it. And so if I have to recruit, and they're all spread out, okay? So it's an equal distribution, but based on population. So Nebraska, South Dakota has the same rate as Dallas, Texas. And so if we have to recruit a certain number of patients for trials, there are geographical considerations just because there aren't enough people in any one center to fully recruit for a trial. So it makes it tough. Secondly, it's a small market. There's only so many users of that drug out there. And so you have private companies who have to justify to their boards and to their stockholders, why would we take the risk and make an investment if our market is so limited? Finally, there's a limited understanding of therapeutic targets. If the biology of a disease isn't understood, and if the natural history of a disease isn't understood, it makes it harder to do the trials. What a company likes better than anything is to have an entity where we know what's going on, we know the implications of the biology, and there's a rationality to their therapeutic intervention. It's now been 9 years ago when the first publication for the anti-aquaporin 4 antibody came out for neuromyelitis optica. This was and still is an orphan disease, but it was lumped with multiple sclerosis for years. And then when that paper came out, not only was there a dramatic shift in focus and research relative to this rare disease because it was biologically interesting. A lot of neuroscientists who've never seen a neuromyelitis optica patient or didn't even know what demyelination was got really excited about the biology and started investing time and resources because there was clear-cut questions to be asked and answered and a path forward to advance our understanding of the disease. Right in line with that, a large number of industries have become interested in neuromyelitis optica, because there's a rationality to the therapeutic approaches that weren't there before. So understanding a disease is incredibly important, and in rare disease, that understanding is often limited. And so the more we can understand the diseases and the populations we treat, the more enticing we are, or could become, to people who will invest resources. So what are the risks to the investments? And this is really important to understand. So the first statistic is 95% of drugs fail to be safe or effective and never come to market. 95% of drugs that are tested at some level. The phases you have to go through to get a drug to market is preclinical testing, which is years of work. The amount of data requested by the FDA, very appropriately, this is not a criticism, I support what they are doing, but the amount of data requested by the FDA before any human being in the United States takes a drug in an investigational status is massive. Everything from testing in tissue culture and cells to doing animal studies, toxicology studies, pregnancy studies at the animal level, and there's section after section after section after section, and it takes years to acquire this data before you can even start to talk about doing a clinical trial. And if at any point in the preclinical, the phase 1, the phase 2, or the phase 3, there is evidence that the drug is either too risky or not effective, it fails. So it won't go further. So there is a huge expense and risk that is taken on by companies to try and bring a drug to market. So I'll take a poll. I don't have the answer on the screen on purpose. What is the current average cost in the United States to bring a drug to market, from start to FDA approval? Anyone want to venture a guess?
750 million.
Higher. Yeah, so depending on how you ask the question, it's between 1 and 5 billion. Okay, billion with a B as in boy, billion. So the number I quote for most is somewhere between 1 and 1.5 billion. I think that's the most accurate assessment. What that accounts, so the $1 billion investment involves everything from the preclinical all the way through the FDA, and then you have to realize that the later you get in this process, the more expense you have, but there's no guarantee of getting it approved. And so you can make a $1 billion bet, go sit in front of the FDA at the end of your phase 3 trial, and they say, yeah, the data's not right. You have to either go back and do another study, or we're just not going to approve this. And that's $1 billion left on the table. Okay? So now you take that risk and you're going to say, all right, I've got a roulette wheel of diseases that I can invest my resources in. Do I want to do diabetes and multiple sclerosis where the payoff on the size of the market is going to justify the risk? Or do I want to do transverse myelitis with 1,500 cases a year? And if you have a very appropriate fiduciary responsibility to your board and your stockholders and your company, this drives a lot of the decision making. So what we have to do as a community is deleverage the risk. We have to make ourselves attractive for the investment. And that's the way the world works today. There is not going to be a large enough infusion of federal dollars in rare diseases ever to solve these issues. We are going to have to partner with industry in different ways to get ourselves across the finish line. So how do we do that? So this is a solvable issue in my mind. So one is biologic understanding. There are many people here in the room who have given of themselves, their data, their blood, their spinal fluid, all sorts of things, in order for scientists across this campus and at Hopkins and at institutions across the United States to better understand the biology of these diseases. Those investments of your time and resources are absolutely priceless. The better we understand these diseases, the more attractive we are to get people with a larger sum of resources to invest in us as a community. Recruitment time. Anything I can do to turn to industry and say, from the moment you say go, I'm going to have a line out the door of willing participants to take part in a study. So instead of taking 2 years to recruit for a study, which means 2 years of expense, that $1 billion, part of it is all the staff it takes to keep a study going. So if it takes you 2 years to recruit, that's expense that has to be paid out with no guarantee. If you can take a 2-year recruitment time and say, we can do it in 6 months, you're a safer bet, because they're not going to have to risk as much funds to get across the finish line. So anything we do at a community level to get people interested in understanding research, and if you are willing to take part, design the study such that it's easy for you to access, easy for you to get to us, easy for us to collect your data and take part in the trials. We can cut our recruitment time, and if we can advertise that to the outside world, again, from our community, we become an attractive area. This is where the TMA comes in. I get a lot of feedback from colleagues around NIH as well as at industry that having a strong patient advocacy group embedded at the grassroots level with people who are invested in actually taking part in research is absolutely invaluable. There's a difference between me putting a study on clinicaltrials.gov and waiting for you to find it, and I can't find anything there, versus Chitra sending out the email saying, or Sandy sending out the email saying, Hopkins is recruiting for this study. If you're interested, please contact them. The engagement at the grassroots level makes a big difference for the recruitment time. And then finally, we need clearly defined outcomes. And this sounds easy. It's not. So for those of you in the audience, some of you have mobility issues. Some of you have pain. Some of you have bladder problems. Some of you have fatigue. Some of you have depression. Some of you have cognitive issues. Some of you have vision issues. Some of you have 2 out of the 3, 4 out of the 5, whatever the case may be. How do I define one outcome measure that measures all of you? Right? So if I enroll 20 of you in a trial for restoration of function, and some of you are walking and some of you are not, how do I score you to prove to the FDA that the drug made a difference? So we have to understand at a baseline level what your issues are, at what prevalence, and in what combination, so we can design a trial that accurately measures you. It's all about metrics. We have to have a number on a piece of paper. You can write every letter you want about how a drug changed your life. The FDA doesn't approve things on that. I have to be able to quantify for the people reviewing this that whatever intervention we did made a difference. And so as we take part in studies that look at defining your outcome, just in a natural state, you were treated, or in some cases not treated, for your transverse myelitis, ADEM, or NMO, what happened to you? What was it, a year later, 2 years later, how were you doing? Such that we can design, if we're gonna intervene, how do we power a study to measure a change?
But that's based on the targeted outcome of the drug.
So if we're talking about repair, the targeted outcome is regaining function. And so, but the function we're going to regain is going to be different for all of you. So we need an ability to understand what happens to the, quote, average transverse myelitis patient, meaning if some people have this degree of recovery on their own versus this degree, what's the average? But what's the variance? How many people don't have as much recovery and how many people have superstar recoveries. How many people here have heard the statistic for transverse myelitis about a third, a third, a third? A third? It's a bullshit statistic. Mike, is that a fair statement? It's a ridiculous statistic. It's based on a really poorly done, long time ago study. It's actually not really a study. And if you ask me, all right, Dr. Greenberg, what's the actual number? I have to say, I think it's blank. I actually don't know because we haven't measured what happens to everybody. And here's one of the problems. If you had transverse myelitis, were mild, got treated, and you are back to normal, you tend not to be at the symposium, right? You're water skiing or, out in Colorado. You haven't filled out a membership form for Sandy, or maybe you did in the first month, but then once you got better, it's in your past. I had my TM 5 years ago. I got nothing. So capturing people as they get diagnosed so we can track what the improvements are. We're doing better than we think. I'm just trying to quantify it. So this is where you come in. It's things like Facebook, it's things like Twitter, it's things like getting connected around the country so we can start to attract people as they get diagnosed. So we can gather their data, gather their specimens, and have a much better understanding of what happens to them is critically important.
Is the complete population of 35,000 BS2?
Yeah, it depends on how you ask it. So another way of asking that is how many of you are there? I don't know the answer because a lot of people who initially get put into the TM category shift categories, either to NMO or to multiple sclerosis or something else. And so it's a hard number to come by. Again, this is where an advocacy group comes in to help us answer some of these questions. So what are the opportunities we have? So there's an ongoing longitudinal study here at UT as well as at other centers. Hopkins has a protocol up and running to track people over time. We don't do anything to you different than what we would do. Okay, so I'm not going to expose you to an experimental drug as part of this study. We are going to ask for you to give data to us on a regular basis. You're going to get phone calls from Sam or from Dina. You're overdue in your questionnaire. Can you please turn it in? And then you've got better things to do, so you forget about it. They're going to call you a week later. You're overdue in your questionnaire. Can you please turn it in? That questionnaire, which is a little bit of annoyance to you or a lot of annoyance to you, is absolutely critical to us answering some of these questions. It sounds silly, but it really isn't. I need to know what's happening to you over time. Are you getting better, staying the same, or getting worse? And I have to put a number around it so I can then go to industry and to the FDA and say, here's what happens to a population of transverse myelitis or NMO or ADEM patients. And if you had a drug that did Y, we could measure the effect. You would see a shift in the curve, but they have to see the nascent data to begin with. The PCORI grant, which we talked about yesterday, and hopefully, if everyone's crossing fingers and toes and everything else, we'll get the NIH longitudinal study funded in the spring. And these are all ways you can take part by giving your data, your specimens, your time, which is very valuable. You have other things to do with life than this. But that sacrifice that you make at any of the institutions goes a long way for us trying to sort this out. This is a way for you as a community to make the community attractive to individuals who can invest significant resources into restoring function. Does that make sense? Okay. So I'm going to get off my soapbox, and we're going to transition.